Yeah, I mean, I think we are one of the first companies to utilize 3D models, organoids and spheroids for therapeutic use. And we are deeply vested in that. And we are targeting the clinical use of organoids and therapeutics in multiple clinical indications. For example, in treating type 1 diabetes, using a pancreatic organoid that we have developed, and it's in the process of being developed. We also have a artificial thymic organoid that will help recover some of the lost thymic function that we see as we age. So I think those are going to be some important progress forward. Again, it's for therapeutic use, not for models. And we are very confident that they will function as intended going forward.
I'm a big proponent of being on the front end of technologies and trying to push the boundaries of what we've been able to do in more physiologically relevant ways. And so 2D formats, as we all know, have been the mainstay for quite some time, decades. 3D models have been out there for decades as well, but they haven't been quite as easily achievable or affordable. And so now I tend to think more in terms of trying to screen not only just tumor models in 3D, but also neurological diseases, neuropathies, and things like that, where you can actually push the boundaries again of the technologies and do 3D screening, small molecule, immuno-oncology, and otherwise, very rapidly and very cost effectively. And that's a big benefit in the recent years, and I expect to see that continue.
I think really the change into 3D models has really filtered down to all aspects. So prior to me joining private sector, I was a faculty member for 12 years at the University of New Mexico, and we started introducing them as a way of sort of what is the real story, what is the real biology, and so we started seeing those filter down more and more. And I think what you're seeing is the use of 3D cultures not becoming the exception, but for many, many labs, it's becoming the norm. And you're learning more and more techniques on how to grow them, how to stain them, and how just to work with them. So I think you're seeing a better use of these 3D models throughout all aspects of science right now.
Well, organoids are at a really nice sweet spot. They're in between a complex in vivo black box animal model and relatively simple 2D monolayer types of cultures that don't really represent the full complexity of an organ. So we're using them a lot. We're using them as a system in which to understand how an organ's phenotype would be affected, and that includes both the disease, but also the basic physiology of the organ. And we're developing new disease models for the different types of disorders that come along with kidney disease or cause kidney disease. So we're using them in almost any application that's going on in the laboratory. They're not an exclusive tool. We also utilize those other types of models, and they're often complementary.
Yeah, so certainly I've been working with a lot more 3D models, organoid models, and advanced models, which I find absolutely fascinating. So I love how my work has transformed in the way that I get to work on these more complex models, just making my work more interesting and feeling more relevant.
So I previously generated a lot of cortical and brain organoids in the lab, and previous to working with those organoids, we used to work with a Huntington's disease mouse model, which was notoriously difficult to work with. And so starting to work with organoids to test our stem cell therapeutic definitely made that testing process a lot easier and a lot more cost effective.